Hi biology students, it's Mrs. Lewis and we are currently in Chapter 8, Section 2, and it's all about photosynthesis. So most autotrophs make organic compounds using photosynthesis. It's the process of converting light energy into chemical energy. And here's the formula. Photosynthesis occurs in two phases. In the light-dependent phase, light energy is converted into chemical energy. And in the light-independent phase, chemical energy used to synthesize glucose. Phase 1, light reactions. The absorption of light is the first step of photosynthesis. Once light energy is captured, it can be stored as ATP or NADPH. Chloroplasts, organelles that capture light energy. These contain two compartments. The thylakoids are flattened sac-like membranes, and the stacks of thylakoids are called grana. The fluid-filled space outside the grana is called the stoma. Light-absorbing colored molecules called pigments are found in the thylakoid membranes. Different pigments absorb specific wavelengths of light, and the most common pigment in plant is chlorophyll. Plants also have accessory pigments. The thylakoid membrane has a large surface area providing space for a large number of electron-transporting molecules and two types of protein complexes called photosystems. These house the light-capturing pigments, and just a reminder, make sure you are coloring in and writing anything that is hard to see in your diagram because your test will be heavily graded on how well you can interpret diagrams. Light energy excites electrons in photosystem 2. This light energy causes a water molecule to split, releasing an electron into the electron transport system, a hydrogen ion into the thylakoid space, and oxygen as a waste product. The activated electrons move from photosystem 2 to an acceptor molecule in the thylakoid membrane. The electron acceptor molecule transfers the electrons along a series of electron carriers called photosystem 1. In the presence of light, photosystem 1 transfers electrons to a protein called ferredoxin. This transfers the electrons to the carrier molecule NADP, forming the energy storage molecule NADPH. Now, at this point, I realize you're probably very overwhelmed. And you have a right to be. This is a lot of information, really hard and really fast, and things that you've never heard about photosynthesis before. And oh yeah, there's that thing again, chemistry from Chapter 6. It can't be helped. We are all on a molecular level. So if you struggle with chemistry and if this sounds like total Greek to you, you really need to schedule some tutor time with me, okay? Don't wait until the last minute because your grade for your final as well as your EOC is going to count a lot for your fourth quarter. ATP is produced in conjunction with the electron transport system through chemiosmosis. ATP is produced through the flow of electrons down a concentration gradient. The breakdown of water provides the necessary protons for ATP synthesis.
The third step is the electron transport chain. This is just a beautiful thing. We're still in the mitochondria and we do require oxygen for this step. This is a very complicated process and we are greatly simplifying it and saying that electrons are transferred from the NADH and the FADH to several electron carriers, and they're used to create a proton gradient. The protons are used to power this amazing enzyme called ATP Synthase. Really, if I could be any enzyme, I'd be ATP Synthase, because this enzyme takes phosphates and adds them to ADP. And if you add one phosphate to ADP, you make ATP. So that's what ATP Synthase does. It makes ATP, and it makes the ATP by adding those phosphates. Now, oxygen, it's the final acceptor of these electrons, and when you have an oxygen molecule combining with two protons, you get H2O, which is water. So water is a product as well. The electron transport chain produces a lot of ATP compared to the other two steps. It's all because of that ATP synthase. There isn't an exact number on this. Many textbooks will say that this step makes 34 ATP, meaning that the net amount of ATP when you added all three steps would be 38 ATP. But you need to understand that this is a perfect case scenario. And in general, you can't really expect that much ATP made each time. Now, if we look at our formula again, we can see how the glucose and the oxygen on the reactant side were used to produce carbon dioxide, a waste product, water, which was a waste product, and ATP energy. ATP energy was our goal. Now, this was just one way of creating ATP energy, a very efficient way at that. But like we had said at the beginning, all cells have to make ATP energy. The way they do it, though, can differ. If there is no oxygen available, some cells have the ability to perform a process known as fermentation.
Phase 2, the Calvin cycle. In the second phase of photosynthesis, called the Calvin cycle, energy is stored in organic molecules such as glucose. The first step of the Calvin cycle is called carbon fixation. CO2 molecules combine with 5 carbon molecules to form 3-phosphoglycerate 3-PGA. In the second step, chemical energy stored in ATP and NADPH is transferred to the 3-PGA to form glyceraldehyde 3-phosphate. In the third step, some G3P molecules leave the cycle to be used for the production of glucose and other organic compounds. And in the fourth and final step, an enzyme called ribosco converts the remaining G3P molecules into 5 carbon molecules called ribulose. These molecules combine with new CO2 and continue the cycle.
Okay, number 2 is the Krebs cycle. This is also called the citric acid cycle. We are now involved in the mitochondria, and this step requires oxygen. The pyruvate that was made before, it's now converted and it will be oxidized. Carbon dioxide, CO2, is produced, and we produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like the NADH, and it will also assist in transferring electrons to make even more ATP.
Alternative pathways, C4 plants. The C4 pathway allows plants to maintain photosynthesis while reducing water loss. Significant structural modification in the arrangement of cells within the leaves separates CO2 uptake from the location of Calvin cycle. CAM plants. Crassulacean acid metabolism, also known as CAM, is found in desert plants. They collect carbon dioxide at night and store it in organic compounds so that during the day they release the carbon dioxide from those organic compounds for the light-dependent cycle of photosynthesis.
We are trying to make ATP energy. Let's take a look at this formula. Remember that reactants, the inputs, are on the left side of the arrow, and products, the outputs, are on the right side of the arrow. This formula, by the way, looks remarkably similar to photosynthesis. I mean, just look how the reactants and the products just seem to be on different sides. This isn't really a coincidence. See, in photosynthesis, organisms like plants and some types of protists, for example, they make glucose. Notice how glucose is a product, but in cellular respiration, we break the glucose. Notice how glucose is a reactant in order to make ATP energy. So, photosynthesis makes glucose, and cellular respiration, it breaks glucose. Kind of cool. Now, photosynthetic organisms, they have the best of both worlds because really, they don't just get to do photosynthesis to make their glucose, they also get to do cellular respiration to break their glucose. I say that's pretty great because glucose is the starter molecule in cellular respiration, and it's needed in order to get all of this going. So if you aren't photosynthetic, like a human or an amoeba, you have to find a food source to get your glucose. Now, cellular respiration involves 3 major steps. We are going to assume that we are starting with one glucose molecule so that you can see what is produced from this one glucose molecule.
The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria.
Cellular respiration occurs in organelles called mitochondria. The mitochondrial electron transport chain is a key component in cellular respiration. The objective of this tutorial is to help you understand the steps of the electron transport chain. All eukaryotes have mitochondria. Let's focus on a plant. Each plant consists of several cells, each of which have mitochondria. A mitochondrion has an outer membrane and an inner membrane. Between the outer and inner membranes is a space called the intermembrane compartment. The inner membrane encloses fluid called the matrix. The reactions of the electron transport chain occur across the inner membrane of the mitochondrion. Respiration is a multiple-step process. Glycolysis and Krebs cycle produce a small amount of ATP and also produce electron carriers called NADH and FADH2. The electron carriers then travel to the electron transport chain, where the electrons they deposit provide the energy to produce many ATP. In fact, most of the ATP that a cell generates is the product of reactions of the electron transport chain. The electron transport chain is a series of 4 proteins embedded in the inner mitochondrial membrane. So let's start our tour of the electron transport chain with the first membrane protein. NADH donates the 2 electrons it received during glycolysis or the Krebs cycle to the first enzyme of the electron transport chain. These 2 electrons will then travel through the second, third, and fourth proteins. Follow the arrow from the 2 electrons at the first enzyme through the other electron carriers. The electrons release energy as they travel through the proteins that make up the electron transport chain. As we will see, the mitochondrion uses this energy to produce ATP. Now, look at the second enzyme of the transport chain. FADH2, which is produced in the Krebs cycle, contributes electrons to the transport chain at this second enzyme. Like the electrons donated by NADH, the electrons donated by FADH2 also release energy as they travel through the third and fourth membrane proteins of the transport chain. Now, look at the fourth enzyme of the transport chain. Since this enzyme is at the end of the transport chain, it has to get rid of the electrons that it's acquired from the third protein in the chain. Otherwise, the whole chain will stop working as the carrier proteins become clogged with electrons. At the fourth enzyme, the electrons are getting dumped onto an oxygen, which is called a terminal electron acceptor. The oxygen then immediately reacts with free hydrogens to form water, one of the end products of respiration. Oxygen's role as a terminal electron acceptor explains why this gas is essential to the survival of most cells. So what happens with all the energy released by the electrons moving through the transport chain? Notice the hydrogen ions that are passing through the first, third, and fourth enzymes. These enzymes use the energy released by the electrons to actively pump hydrogen ions from the matrix to the intermembrane compartment. This process creates a hydrogen ion gradient, also called a proton gradient, meaning that the concentration of hydrogen ions in the intermembrane compartment is now higher than it is in the matrix. Now, focus your attention on the ATP synthase protein at the bottom of the figure. If you follow the black arrow through ATP synthase, you'll see that the hydrogen ions enter ATP synthase from the intermembrane compartment and move through it to the matrix by facilitated diffusion. Since the hydrogen ions are moving from a high concentration to a low concentration, they're going